This is Biology 1, Chapter 12, Part 6 on molecular genetics. In this portion of the video lecture, we will be taking a look at gene regulation and different types of mutations. Genes need to be regulated. They need to be essentially turned on and turned off at the right time. Just like the cell cycle, gene regulation also needs to go 100% correct 100% of the time. Prokaryotes. Remember, prokaryotes are very simple organisms such as bacteria. They do not have a nucleus and several organelles. Gene regulation, prokaryotes can actually control their genes in response to what's going on around them in the environment. And they use what's called an operon. An operon is kind of like an operator. It connects genes for a metabolic pathway. So like way back in the old days, when you had to call the operator to connect your phone line to the person you wanted to call, that's what the operon does. It switches genes on and off when they need to be. One type of operon is the tryptophan operon, or TRP operon. When the TRP operon is on, then the enzymes for making tryptophan can be made. If that gene is switched off or repressed, then those enzymes are not made for protein or for tryptophan synthesis. So when the TRP operon is on, we can get tryptophan. When the TRP operon is off, no tryptophan is made. The lactose operon, or the lac operon, controls production of enzymes to digest lactose. So again, when the lac operon is on, those enzymes for lactose digestion will be made. If the operon or operator is switched off, the call can't be connected, so the enzymes for lactose digestion will not be made. Now, eukaryote regulation, we are, as eukaryotic organisms with the nucleus and organelles, and multicellular, we are complex organisms, and we need our genes to be switched on and off at the right time too for successful gene regulation. In most animals, if you think of the DNA as like a blueprint for the physical aspects of an organism, the Hox genes would be those genes that are responsible for making limbs and eyes and stomachs and muscles and all of those things that are in the general body pattern of most animals. Hox is short for homeobox genes. These genes are called homeobox because they are similar in many animals, and the directions in the homeobox genes, or the Hox genes, are those genes responsible for making the blueprint of the organism, the body plan of that organism. If mistakes are made in the Hox genes during the decoding process, there could be some very interesting functional problems with that organism. These are some fruit flies that have had their Hox genes messed with a little bit. In the different types of alterations, they call them, you can make wings look blistery or wrinkled. You can give extra legs if you would like. You can also replace and switch Hox genes. You can take, for example, the Hox gene that is responsible for an antenna and place that in the area of the homeobox where it has information to make a leg. And in the resulting organism, you will get a leg growing out where the antenna should be, and get an antenna where the leg is, where the leg should be growing. So Hox genes are the general body plan of most animals. If mistakes are made there, there's a large problem functionally for the organism. Mutations. A lot of common misconceptions about the word mutation. Usually, most people think mutations are something that happens to you sometime in your life, like the Teenage Mutant Ninja Turtles or the X-Men, and somehow, because of an environmental exposure, that you have been mutated because of that. And it's always bad. Mutations are not bad, inherently. Some mutations can actually be good, and many mutations don't make a bit of difference in an organism. It all depends on where that mutation is taking place and how that mutation occurs. But a mutation, any kind, good, bad, or indifferent, all it is a permanent change that occurs in a cell's DNA. And there's different types of mutations. A point mutation is where at a certain point along the DNA molecule, there's been a chemical change in one base pair. Types of point mutations. Sometimes you exchange one base for another. So just like a substitute teacher comes in and teaches a class while the regular teacher is gone, same idea. One base is being substituted for another. A missense point mutation is for some reason that the transfer RNA grabs the wrong amino acid, and the wrong amino acid is coded for. In a nonsense point mutation, there is some reason along the point of the DNA molecule where translation, or making amino acids into proteins, just stops for no reason. So stopping translation early is called a nonsense mutation because it makes very little sense. Here's an example of a point mutation. Again, the Hox genes, those animal body plan genes. You can see here in a normal fruit fly, the antenna, where it should be, on the left, and then on the right, you can actually flip-flop. It's almost like Mr. Potato Head with Hox genes. You can take the Hox genes for a leg, stick it in the area of the DNA that's supposed to be making an antenna, and you can grow a leg out of your face if you're a fruit fly. Lots of point mutations will cause different organisms to behave differently. Here we have a frog with a multiple number pairs of legs. Sometimes those point mutations, like for example the nonsense, when things stop early. This is an example of a nonsense mutation where the leg wasn't, or both legs, were not made correctly because that information just stopped. A frame shift mutation is a little bit different than a point. Frame shift mutations are causing the 3-base code to shift to the left or the right, depending on if extra bases are added or some are accidentally taken away. Insertion. Okay, when you insert, you're going to add an extra base. Deletion is where you're going to lose a base. And duplication is adding an extra duplicate base. What will happen is, because codons are read in a 3-letter sequence, when you start adding or taking away bases, that's going to cause the reading of the amino acid chain to be different. And that can cause some problems. Albinism is a type of frame shift mutation. About 1 in every 64 alligators are born as an albino. Out in nature, of course, they don't live to be as old as this particular alligator, because in general, if you have no pigment and you are in a green, brown, foresty, swampy sort of area, you're going to stick out like a sore thumb, and you'll be picked off by a predator before you can get to be this age. This guy resides in a zoo in the state of California. Here's another type of albino mutation. This is a corn snake. This is actually a snake that is a friend of mine. The snake's name is Five, so when you see him, you can say, "Hi, Five," and put your hand up against the cage, and he'll actually put his face up there and give you a high five. Frame shift mutations can also cause some problems in Hox genes. This was a kitten. Unfortunately, it did not make it to adult life. The problem with this kitten was that there were 2 mouths and 2 different esophaguses or throats, but it only had 1 stomach. And unfortunately, and it also shared a brain. Unfortunately, this little guy kind of ate himself to death. But again, some mutations can be lived with and some can't. But not all mutations are bad. Expanding mutations. These are going to add repeating nucleotides for every generation or every new cell that's made. And we call these types of expanding mutations tandem repeats because every generation gets 2 or more repeated bases every time the cell divides. So it's like repeated information over and over and over again. Here's another example of a mutation. Most mutations are usually what are called a mirror image. So like the kitty cat example, you'll get a left to right sort of mutation. In this case, with this little guy, this turtle, which survived until adulthood, they have more of an up and down type of mutation, where it's, instead of left to right, it's kind of top to bottom. This cow has another kind of side-to-side mutation. There was only one functioning eye and nose and mouth. The other portion was not a fully developed individual. And again, unfortunately, because of the difficulty this little cow had in getting its nutrients, it did not live very long either. So here's how you can take a look at how the cell would read mutations. If you could make a sentence with every letter being 3 words long, just like a codon, you can make a complete sentence. So for example, up at the top, the sentence is, the big fat cat ate the wet rat. Now, if a cell is reading this information or you're reading it, you understand what's happening. There's a cat, it's big and fat, and it ate a rat that's wet, okay? If you substitute in a missense the G, for example, if you substitute that with a Z, now you have the biz fat cat ate the wet rat. Now in general, the cell still understands what's going on, but biz is not a word. A nonsense mutation. Remember, that's when something stops early for no reason. So you went from the big fat cat ate the wet rat to the big rat. Now there's a lot of information that's missing there. Deletion. Here's a frame shift example. If you take out of the first word in the sentence, if you delete that E, what the cell will read is everything shifted to the left. So instead of the big fat cat ate the wet rat, now we have the cell does not know what that means, okay? So that's what it means by a frame shift, is everything is shifted over. It's still in 3 letters because the codon, those codons only work in 3 letters. So if you boot one out, if you delete one, everything's going to shift. Likewise, if you insert a letter, so if you insert the letter Z in front of the word fat, now what you have is the big again. After that point, there is no more understanding of the cell of what's going on because it doesn't recognize those codons or those words. A duplication. Here's an example of that. We duplicate the codon for fat. Now we have the big fat cat ate the wet rat. In general, the cell understands what's going on, but that repeated information is not necessary. The tandem repeats, these expanding mutations. Here we have, again, the big fat cat ate the wet rat. We can keep going on with the cat, and every generation that comes, you end up with more repeated information. Protein folding and stability. Okay, everything's got to go 100% correctly, 100% of the time. If they're not, the protein won't fold correctly. And if proteins don't fold correctly, that protein is defective or it will malfunction. Mutagens are that environmental exposure to cancer. Mutagens cannot be passed on from generation to generation. Mutations in body cells. If I somehow develop skin cancer or melanoma because of exposure to UV rays, my offspring will not have melanoma cancer. However, if I have a mutation in my egg cells, that will get passed on to the next generation. This has been Chapter 12, Part